thanks very much. First, I'd like to thank the organizers for inviting me to give a talk in this nice workshop. Very diverse, very interesting talks. I enjoyed it very much, and more to come, I guess. So I'd like to talk about synthetic biology of antibiotic production. And as you all know, and this has come up in some of the talks already, there's a big problem with antibiotic resistance. It's an increasing problem as well. So in hospital infections, there's 3 million patients per year in the EU, 50,000 deaths, and 25% of those colonized in hospitals have subsequent infections. And this resistance is increasing. And you can see from this figure here, MRSA increasing massively. Even resistant enterococci to vancomycin, so the vancomycin is the so-called drug of last resort, is also increasing as well. Now, if you look at the different EU countries and proportion of MRSA among invasive Staphylococcus aureus, you can see these red countries, which includes the UK and Ireland, France, Spain, Italy as well. Well, there is a problem with MRSA. Now, I come from the Netherlands, and the Netherlands is this light green here in the middle of this red and the orange bit. And I was thinking, why is it that the Dutch don't have so much problem with MRSA? Well, first of all, the Dutch like to cycle everywhere. They don't use cars very much. It's everyone, not just the younger people, but people who commute, adults who commute, go by cycles all the time. Pensioners also are cycling all the time, so they're quite healthy, in fact, and they rarely go to the doctors even if they're ill. But if they are ill, and if they do have to go to the doctors, it's a GP system, just like in the UK. So if you call up the GP, the assistant that picks up the phone will tell you, you don't have to come. I've had this experience several times. I had to fight on the phone to be able to see a doctor. And then once you do see a doctor, the doctor will not give you anything. He'll just tell you to go home. So basically, they don't really prescribe antibiotics that easily. You have to be pretty ill. The other thing is, if you are ill, you're out of the country and you do become very ill, what they'll do is quarantine you for at least 2 weeks so that you don't bring it into the country. And I think these are some of the aspects why Netherlands is quite low in the MRSA proportions. Now, if you think of antibiotic discovery, okay, penicillin was the first to be discovered. Here's a map of all the antibiotics, commercial antibiotics that's on the market now and when it was discovered. So starting from 1940s, there's a peak around 1960s, but you can see there's a massive decline. And actually, there isn't a lot of novel commercial antibiotics being found at the moment. And we wondered why. Okay, some people say there is absolutely no novel antibiotics out there in the nature to be discovered, which I totally disagree, and we'll come back to that in a minute. Another reason, perhaps, I was thinking was the pharmaceutical industry has stopped natural product discovery. And there's probably a reason why. So any drugs that need to be on the commercial market has to go through clinical trials, and it costs a lot of money. Even if it's antibiotics or if it's anti-Alzheimer or anti-cholesterols. And of course, anti-cholesterols and anti-Alzheimer, so you take it for as long as you live. But for antibiotics, you take it for 2 weeks, and that's it. You don't take it anymore because you should be cured. So the profit that the pharmaceutical industries get is quite low for antibiotics, and perhaps that's why they're not really trying to look for new natural products anymore, because it doesn't make much money. But of course, we have problems with resistance, so we do really need to find novel antibiotics. So how can we find them? The traditional way of looking for them in companies as well is to use strains that they already have, screen them for doing mutagenesis, screen them in different conditions, and see if they can find other antibiotics being produced. Or you can look for very rare Actinomycetes, even in soils outside the university, for example. You can find very rare Actinomycetes. Some might look for, do some metagenomics. Some might look for strains in very extreme conditions, like in very cold places, Arctic, under the sea, hot vents, so on and so forth. But in fact, all these things have been done, and we still don't find a lot of novel antibiotics being found. So what do we want to do? Well, I would like to propose that we can do synthetic biology to try and find novel antibiotics. And Before I go into this, I know that we had a very nice talk on artemisinin, and you don't need to explain this slide at all, but I just want to make one point about this synthetic biology. So what I'd like to do is take this engineering one step further. So what Amaris and Jay has done is to make a natural product which is already around in a different host. What I would like to do is to make a product that nature has not been able to see at all, a really novel compound or structure. And how do we do this? Well, for this I need to explain a little bit about how antibiotics are biosynthesized. So this is a scanning EM picture of Actinomycetes. Here's the spores, here's the soil here. And Actinomycetes are the main producers of antibiotics. About 80 to 90% of commercial antibiotics are produced by this species. Here's a colony taken from the top of Streptomyces, and you can see these blue blobs. They're all antibiotics being secreted out from the colony. Here's a cross section, if you cross it this way, of a colony, and you can see the inside where there's a red pigmented antibiotic being accumulated in the cell. And here are some chemical structures of different antibiotics. Here's penicillin, here's erythromycin, this is vancomycin, which we just talked about, and we have very big compounds as well. But now, how all these compounds are biosynthesized is in fact very similar to each other, even though the structure can look very different. And the way it's synthesized is like this. So we have big open reading frames like this. This is in the case of erythromycin. Within the big open reading frame, we have modules. Within the modules, you have domains, these circle blobs. And these domains are where the enzyme activity occurs. And in the case of erythromycin, what happens is there's a starter unit which is using C3 units. It's loaded on here, and then in this module, another C3 unit is extended, and then another C3, and so on and so forth to make a long chain. So it's very similar to the fatty acid biosynthesis. So at the end, there's an enzyme that stops the enzyme, cleaves it off the whole long thing, and cyclizes. There's other modifying enzymes, and you get your erythromycin. Now, if you change this C3 units to peptides, you get something like this. Again, big open reading frame, modules, and then domains. And on these domains, these annulation domains, specific amino acids are targeted to be loaded and elongated. And you get your long chain again, your thioesterase to cleave it off and cyclize, so you get your compound. Now, the most important thing, the characteristic of this biosynthesis is that, as you can see, it's modular. Because it's modular, you can quickly see that how we could change these modules around, or even the domains, to be able to create lots and lots of chemical structures, novel structures. So exactly how do we want to do this? Well, we'd like to identify all secondary metabolite gene clusters in public databases, genome sequences, even from private genome sequences. It doesn't matter where it comes from, bacteria, plants, whatever. We also like to identify enzymes that may have similar enzyme activity to some of these biosynthesis enzymes. We can even take enzymes and actually change their substrate specificity or even their activity as well. We can redesign them. And then we'd put these genes together in one transcriptional unit, put a synthetic promoter in front and a synthetic ribosomal binding site, and put these units, several of these units, to make a compound. And we'll put this onto a clickable vector like this, so we can put this into a screening host. And once we screen for novel antibiotics, and once we have found the novel antibiotic, then we can put this whole thing into an expression host. And why this? Because the expression host will now have the primary metabolism and the flux geared to produce a lot of antibiotics. So we'll have a lot of these compounds at the end. Now, we need a lot of computational tools as well to do this. So for example, if we say, okay, I want to make a specific compound, you have to predict some of the pathways. Of course, there are these tools here, software tools one can use to predict them. And one has to think about availability of enzyme activities, thermodynamic constraints. But if you have too many predictions, of course, you have to prioritize the pathway. So to do this, okay, you think about fewest reactions, best understood chemistry, no toxic intermediates, and one more thing is to do metabolic modeling so that we can look at host suitability and sustainability of flux. And then we get to the pathway that we've been able to select. And after that, of course, we have to do the refactoring. This is where all the heavy workload comes in. And of course, there are softwares here to guide us with that as well. Now, synthetic biology is not just about refactoring. In fact, we have to think a little bit more about other things as well. For example, these proteins we need multiple proteins to actually make this end compound. So we can think of putting synthetic protein scaffolds together so the enzyme activity is more effective. We can think of making compartments. Now, yeast and filaments and fungi already do have compartments, and they do produce antibiotics in these compartments. But why not in bacteria? We can make compartments in bacteria. And again, to make enzymes more efficient in their activity. We can have different microbial consortia, so have one microbe producing a specialized precursor, and the second actually making the end product. So we can also think about transcription, okay? How do we control it? We can control it so that it's just, you only have the expression when you need it. So we can control it by allosteric control. This is very fast. So the first enzyme end compound activating the second enzyme, and so on and so forth, very quickly. If you want it a little bit slower, we can think of a just-in-time gene expression where the transcripts are only expressed exactly when you want them. Now, unicellular organisms we know that are quite heterogeneous as well. So we need to really synchronize each of these cells at their specific stages. And this is quite important, especially for production, antibiotic production. So we can think of using signaling molecules to make absolutely sure that each of the cells are exactly at the same phase of growth. We can also think of using signaling molecules to just express one transcriptional unit. For example, the first part where the core enzyme structure is made, and then express the modifying enzyme units. We can think something like this. So what do we exactly need? These were ideas. So what do we exactly need for synthetic biology of antibiotics? So first of all, like I said, we need the biosynthesis gene clusters. We need a library of building blocks from different sources, whatever sources, sequences you have. One needs regulatory circuits to control gene expression. You need a lot of computational analysis to do systems modeling, simulations, also to look at flux balance analysis. One also needs vectors because these biosynthetic gene clusters are very big. There can be up to 50 to 100 kb. So we need stable vectors. We also want to control the gene expression, not on transcription level, but also on translational levels as well. And of course, we need to engineer the chassis, the host, so it is geared up to whatever antibiotic production you want. And of course, you need to debug the routines. So in the next few slides, I'd like to show you what our group is doing on each of these steps. So first of all, what we've done is to produce a software called antiSMASH. So here's a website link for this software. And what it does is it's a rapid genome detection annotation of secondary metabolite biosynthesis gene clusters. You can put whole genomes in there. You can put just parts of genome sequence if you have into this. This is web-based, but if you don't want to use the internet, you can also download this software onto your computer so you can do it in-house as well. And this is an example of what you'll see after you put a genome sequence in here. In fact, it's detected 24 different, this type of secondary metabolite gene clusters, and we're clicking at number 10. It shows you the gene organization, and in these core gene structures, you can see the domains, and it even gives you a predicted core structure as well. So using this software, what we've done is to analyze Streptomyces clavuligerus genome. Now, Streptomyces clavuligerus is a commercial producer, and it's not an antibiotic, but it's a producer. It produces beta-lactamase inhibitors. So if you take any beta-lactams, this will always be in there. So the surprising thing that we found from this genome sequence was that it has a very small genome. It's about 6 megabases. All of the Actinomycetes have up to 9, 10. So compared to that, it's very small. We also found that it has a huge linear plasmid. It had 1.6 megabase linear plasmid. But even more surprising was that we found close to 50 secondary metabolite gene clusters. Now, this strain is known to produce about 5 antibiotics with known structures. So 45 of them, the rest of the 45, has a potential to be a novel antibiotic. And we're trying to express these so-called sleeping genes. And we've done this as a proof of concept from a different Streptomyces species called Streptomyces silicolor. And again, we found an orphan gene cluster. We call it orphan because we don't know what the end product was. The end product was never known. However, we do know that these proteins were expressed, synthesized. So what we've done is to delete this regulator here, and the mutant now produces a yellow color. This is the wild type here with the blue, making blue and red. And we've never seen this yellow color before. This strain was worked on for about 20, 30 years. And up until now, we've never seen this yellow color. So it was a revelation to us that actually we've been able to awaken this orphan gene cluster. And in fact, a bonus for us was that it has antibacterial activity. So this is a proof of concept, but we know now that there's several Actinomyces genomes being sequenced, and it's not atypical from Streptomyces clavuligerus. All of these Actinomyces genomes do have 20 to 30 secondary metabolite gene clusters identified. So it's not that there isn't any more antibiotics out there in the soil, we just didn't have the way of finding it. It's all there in the genome. So what my group is doing is using synthetic biology to actually awaken all these secondary metabolite gene clusters that has not been awakened yet, to look for novel compounds, of course. So this is something that we've done quite recently, very preliminary, so it's just a sneak preview kind of thing. We're trying to identify building blocks as well by using many. We have hundreds of secondary metabolite gene clusters now, and using those, we've been trying to identify building blocks. Sorry. So what's that? Well, it means that there's several genes that seems to be involved in a specific chemical moiety to build up a special chemical moiety. This is some example. So chlorobiosin is this compound here, this N compound. This is the biosynthesis genes for chlorobiosin, and these 4 genes here are responsible for building this chemical moiety. Here's coumaromycin, A1. Again, here's the structure, and again, the same 4 homolog genes are responsible for those chemical moieties. Novobiosin, again, the structure here, the biosynthesis gene, and again, 4 genes, all needed to make this chemical moiety. These 3 are in the same class of antibiotics called aminocoumarins. Now, this one is not. But you can see that in the chemical structure, here's a very similar chemical structure like this. And in fact, we found, in this case, it's not clustered together, and in this case, it's only 3, but we found the homologs in this biosynthetic cluster. So this means that we can look at, if we want to build a specific chemical moiety, then we can go and use this and plug this into our pathway and know that it'll probably produce this chemical structure. Another added on value to this is if you've got antibody biosynthesis cluster and you don't know what the end product is, and you do find a cluster of genes like this, you can predict it probably has this structure in there as an compound. And you can do the reverse. If you have a compound and you have this structure, you can guess in the biosynthesis gene cluster that it'll probably have these genes there as well. So what else do we need to make a library of parts? Well, we've talked about the one before, but we were also making other softwares to actually look for operons. So we know that a lot of the genes are conserved in operons, so we're looking for them for orthogonal operons and put them into our library. Of course, one can do blasts to look for enzymes that are similar to one another, but you can also look at 3D structures of enzymes and look for enzymes that may have identical substrate specificity and put them into the library. And of course, these things also we can use so that we can change, in fact, the substrate specificity to make it into library parts. And together with a library of promoters, we're actually starting to create this using T7 promoters in Streptomyces, and also with ribosome binding sites, which have all different strength. You can start now to put together a pathway. Of course, first we could do in silico testing, and then actually doing in the actual experimental parts as well. So what else do we do? Well, we're also looking at regulatory circuits. This is in Streptomyces. There's small molecules called gamma-butyrolactones, which are involved in antibiotics production. So what it's shown here is, this is the chemical structure of the butyrolactones here. What we show is that if you isolate these butyrolactones from different growth phases of the Streptomyces silica oligo growth and spot them onto an indicator lawn, you see these halos starting to produce. And that's because of these signaling molecules actually affecting or making precursors antibiotics. We know how the genes that are involved in synthesis, we know the receptor, and we're trying to understand the fine details of the signal transduction pathway for this small signaling molecule. But in terms of synthetic biology, what I'd like to do further is to use this receptor, re-engineer it, and also re-engineer the signaling molecules so that we can use this whole circuit as an orthogonal system, a regulatory system. And we can also use this system for synchronizing molecular clocks, like I said before, and also to express different transcriptional units at different time points. We're also creating novel switches like ribo switches and circuits as well. We're also working on antisense RNA, non-coding RNA, to use to control protein synthesis. So Rainer talked about this yesterday a little bit, but what we've done is to find an antisense gene in glutamine synthetase. It's actually in the open reading frame, so it's 100% complementary. So it's a little bit different to how it works in normal bacterial systems. It's more closer to the eukaryotic system. So what we've done here is to overexpress the antisense glutamine synthetase genes, and you can see here, this is a Western, that the protein synthesis has gone down a little bit when it's induced. So we can use this method also to control protein synthesis, especially for those genes where it's toxic, where you can't really delete them. We're doing a lot of computational analysis as well, one on the SCB on regulatory circuitry, the regulation system that I just spoke about. So from this modeling, what we've been able to find is that it's indeed this whole regulatory circuit is a bistable switch for antibiotic production. We're also doing constraint-based metabolite modeling to try and predict expression host suitability. So what we've done is use close to 40 Actinomycetes metabolite models, and into them we've put in 5 different classes of different antibiotic biosynthesis pathways, and then asked which hosts would be great to use. And the light colors means that they're very good in producing the antibiotics, and the dark colors mean they're not. And it's quite surprising because there are some strains like here, which is quite okay to produce all classes of antibiotics, while some of them, like down here, is not good for any. And these, I'm sorry that you can't see this here, but are not Streptomyces, which are the strains known to produce antibiotics. Actually, these are Mycobacterium species. So this is very interesting. This is, of course, only a prediction. So now we need to do the wet work to see if this really works or not. We're also doing some flux balance analysis to use to redirect flux. So what we've done is to model the Streptomyces clavuligerus metabolite and then use microarray analysis data to predict which enzymes we can get rid of. So here, in the red are all the enzyme pathways that we do need, primary enzyme pathways. But in the green are those that we can get rid of. So this is a way of trying to reduce the genome as well, so it's more compact. So we're also doing some engineering of the chassis, making minimal genomes like the way we were talking about, reducing getting rid of any of the enzymes that we don't need, and also using this clavuligerus genome, which is very small compared to others. We're trying to make stable vectors to be able to insert large inserts, and of course, engineering primary metabolite pathways. Another thing that I'm currently doing as well is trying to make compartments in bacteria. And this is not a lipid-based compartment, this is a protein-based compartment. So this is a protein-based compartment using the ethanoamine utilization genes. And this is already published, I think about 2 months ago, that people have been able to put GFP into this compartment in E. coli. So our idea would be to put antibiotic biosynthesis gene clusters into this compartment. Small molecules can go in freely, but large compounds cannot. So it can stay into the compartment. For example, if it's a toxic end compound, you can keep it in here. Another thing, of course, what Rainer talked more in detail about yesterday is about debugging routines and doing untargeted metabolite analysis. And here, he's already talked to you about the non-coding RNA, using non-coding RNA, and looking at metabolic change. This was a proof of concept for using this LCMS for studies like this. But we're also using this LCMS methods to look for small molecule structures and to identify the structures. So I hope I've been able to show you that we're actually developing lots of tools to produce novel antibiotics for its discovery and design. But in fact, it's not just for novel antibiotics, it's for any functional metabolites. We can extend it further, and that is our goal in future, to extend this much further, not just staying into antibiotics, but any kind of functional metabolites. So last but not least, I'd like to thank all the current members of my group, and also the past members in my group who has contributed the work here. This work cannot have been done without a lot of collaborations from different disciplines. On the top here are all bioinformatics people. Rainer mainly doing bioinformatics on metabolite analysis and also modeling. Michael and Tillman helping us with the anti-SMASH. Wei Shu helping us with the modeling of the regulatory systems. We have a 4-group consortium which we're trying to actually look for novel antibiotics, which includes 2 companies, DSM from the Netherlands, Catetogen from Italy, a group in from University of Amsterdam who are specialists in screening, and also Christian from Hans Knoll Institute in Germany who does chemical structure analysis, and also Craig Venter Institute for the sequence of clavuligerus, and also collaboration with Osaka University from Taka Nishida, we're collaborating on the regulatory system of Streptomyces. And also I'd like to thank all the funding bodies, and also thank you for listening to the talk. Thanks very much.